Welcome, everyone, to our educational webinar entitled hPSC-Derived Neural Crest, Induction and Axial Patterning. My name is Teresa Grieco. I'm a scientific marketing specialist at STEMCELL Technologies, and I'll be your moderator for this webinar. We are excited to have Dr. James Hackland as our guest speaker today. Dr. Hackland is a research fellow at the Department of Developmental Biology at Memorial Sloan Kettering Cancer Center in New York. He trained in stem cell biology at the University of Sheffield with Dr. Peter Andrews, specializing in stem cell differentiation to neural crest. He then went on to identify the role of key signals involved in trunk axial patterning of hPSC-derived neural crest during his postdoctoral work at University of California, Riverside with Dr. Martin Garcia Castro. In collaboration with Dr. Lawrence Studer at the Sloan Kettering Institute, Dr. Hackland uses his expertise to study the biology of the neural crest and establish it as an in vitro model towards cell replacement therapies. Let me now hand you over to Dr. Hackland to begin the presentation.
Thanks for the introduction, Teresa, and thanks for everyone for joining me today. So I've designed this talk to give you an overview of the human pluripotent stem cell-derived neural crest field with a specific focus on induction and axial patterning. I'll be talking partly about my own work, but also discussing other notable contributions to the field. The image I'm using for my title slide depicts sensory neurons expressing islet-1 and peripherin that were generated from human pluripotent stem cell-derived neural crest. So I'll start today by introducing the neural crest, first from an evolutionary perspective and then from a developmental perspective, with a tiny bit of history thrown in. After that, I'm going to move on to a discussion of the signals that are required to induce human pluripotent stem cells to differentiate into a neural crest-like state in vitro. This process has been developed over the course of the last 15 years or so, and I'll discuss how the field has advanced over time. Then I'll go into detail about one of those signals in particular, BMP, which was a main focus of the work that I did in the Andrews lab as a graduate student. After this, I'm going to move to axial patterning. The neural crest forms down the axis of the embryo and exhibits a restricted differentiation potential according to the position at which it forms. For this reason, it's important that we're able to derive neural crest-like cells that are functionally equivalent to those in vivo at these different axial levels. I'll discuss the signals that are involved in the specification of these different axial identities, and then finally, I'll talk a little bit about applications for human pluripotent stem cell-derived neural crest. So the neural crest is a synapomorphy of vertebrates. So that means that it's a vertebrate innovation and as such exists in some form in all vertebrates. Vertebrates are chordates along with two other groups or subphyla, cephalochordates and tunicates. So initially it was actually assumed that cephalochordates were the most closely related sister group of vertebrates, and this was based largely on the macromorphology of the extant species within the group. However, more recently, phylogenetic analysis and the discovery of neural crest-like migratory cells by William Jeffrey at the University of Maryland have shown that tunicates are likely to be more closely related to vertebrates. The neural crest-like cells that exist in tunicates delaminate from the neural tube and migrate to the surface of the organism, where they differentiate into pigment cells, but they actually don't generate any of the other derivatives seen in vertebrates. Brian Hall and others have made the argument that the vast array of different cell types that are derived from the neural crest in vertebrates, which is actually more than from the mesoderm, means that the neural crest should be considered a fourth germ layer and vertebrates quadrablastic. And you'll often hear this in reviews and introductions. Evolutionarily speaking, the neural crest appears to have acquired the ability to generate these different derivatives through a combination of innovation and co-option of existing developmental programs. So examples of these two paths are dentine producing odontoblasts, which are restricted to the neural crest and an example of innovation, and chondrocytes, which can also be derived from the mesoderm and are evolutionarily speaking older than the neural crest. Even with invertebrates, there's a great deal of variation in the differentiation potential of neural crest. the clearest example being the division between jawed vertebrates, gnathostomes, and jawless vertebrates, cyclostomes. The jaw and much of the craniofacial mesenchyme of gnathostomes is actually neural crest derived, and the evolution of this is thought to, at least by some, to have enabled the gnathostomes to adopt a more effective predatory lifestyle than their ancestors. The discovery of the neural crest is credited to Wilhelm His, who identified it in chicken embryos using a newly improved microtome that enabled him to make micrometer advances and thereby take very thin sections. He used the term intermediate cord or strand rather than neural crest to describe a structure situated in between the developing neural tube and presumptive epidermis that he identified as the origin of the cranial and spinal ganglia. Although in this work His didn't identify any other neural crest derivatives, he allowed for the possibility that there could be more by saying whether the entire intermediate cord is used up when the sensitive ganglia are formed must be viewed as an open question. The term neural crest was actually coined by Arthur Milnes Marshall, who was a student of one of His's contemporaries, Francis Balfour. So the middle figure I've taken from His's 1868 publication, and it shows a series of progressively posterior sections of what he described as a stage 5 chicken embryo. And this is actually roughly equivalent to an Hamburger-Hamilton stage 8 embryo, which is how we'd refer to it today. And you can also see this stage sketch as a whole mount by His on the right-hand side of the slide. Although each of these sections are from the same embryo, they show different stages of neural tube closure. And that's because this is a process which occurs down the entire length of the embryo in a temporally segregated manner and sort of zips up, at least in the trunk, not unlike the closing of a zip. Using the comparative embryological techniques of the time, His was only able to identify the neural crest post-delamination, and debate continued into the 20th century as to whether the neural crest emanated from the neural tube or developed as an entity unto itself. We now know that not only does the neural crest form in a neural plate border region that's distinct from the neural plate, but that the early specification events for this tissue actually occurred during gastrulation, long before delamination. So here you can see two more of His's sketches of a gastrulating chicken embryo at what we'd now refer to as HH stage 4. A 2006 paper by Bash and Garcia Castro was able to show that a lateral portion of the chicken epiblast at this stage, around where I've put the epiblast label, is already specified towards a neural crest identity. They did this by dissecting this region and culturing it ex vivo in neutral conditions, where it was subsequently observed to upregulate the neural crest specifier PAX7, while adjacent regions didn't do this. They were also able to show that this process of specification could only occur in the presence of both Wnt and BMP signaling, as inhibition of either of these two signals blocked the formation of the tissue. So this finding corroborated a lot of the existing evidence of a requirement for these two signals, but it highlighted the early stage from which they play a role. And I draw attention to it because, as we'll see in a couple of slides' time, both of these signals are key in inducing human pluripotent stem cells towards a neural crest fate. So speaking of human pluripotent stem cells, it was actually just a year before that publication that the first article describing the differentiation of human embryonic stem cells into neural crest-like cells and sensory neurons was published by Oz Pomp and Ronald Goldstein. So here you can see that the neurons they generated were positive for the sensory neural marker BRIN3A, as well as peripherin, a marker which is restricted to the peripheral nervous system. In order to generate the neurons that you see here, Pomp et al. Co-cultured the human embryonic stem cell line HES1, as well as a couple of other human embryonic stem cell lines, alongside an inactivated mouse stromal cell line known as PA6. This is a technique that utilizes the so-called stromal-derived inducing activity, or SDIA, that in this period have been used successfully to generate peripheral neurons from mouse ES cells, as well as central nervous system neurons from both mouse and human ES cells. Using this approach, it took them about 3 weeks to generate peripheral neurons using media first supplemented with knockout serum and then subsequently with N2. After approximately a week, they observed the upregulation of neural crest-associated transcription factors, snail, hand, and SOX9 by PCR. This work showed that just like mouse ES cells, human ES cells could also be directed towards a peripheral neural fate. And it also demonstrated that neural crest-like cells can be generated from human embryonic stem cells in just 7 days, when it takes just under 3 weeks for the tissue to appear during human embryonic development. So I've expanded this schematic to show a summary of the key neural crest induction protocols that have been developed since the PUMP publication. So along the top, you can see the date that the protocol was first published, and down the side is the amount of time it takes in days for neural crest cells to be generated. The method used to start the differentiation, whether co-culture, as with the PUMP publication, confluent layer of embryonic stem cells, aggregates, or single cells, is represented in the sketch at the top of each protocol. The color of the downward arrow represents the media supplement, whether it's knockout serum, a BSA-containing supplement like B27, or a BSA-free supplement like N2. And the signals used to direct the cells towards a neural crest fate are represented by colored bars to the right of the arrow. So as you can see, the PUMP publication kicked off a 15-year-long period of refinement that is depicted here and resulted in a move towards better defined systems. No more co-culture and SDIA, a gradual move away from knockout serum, and even from BSA-containing approaches. There's also a move away from purification, whether manual or by fax, and this is because gradually these publications generate neural crest with a higher efficiency and less neurolectoderm contaminating the cultures. This process also resulted in the identification of a number of key signals. There is a clear requirement for a lack of TGF-beta signaling, and in most cases, the small molecule SB431542 is used to inhibit TGF-beta. In 2011, there was a publication by Menendez et al. That showed that stimulating Wnt signaling, either with recombinant Wnt or with a GSK3 beta inhibitor, they used bio, greatly improves neural crest induction efficiency, and this has been adopted subsequently in the other protocols shown here. And then the last signal that I want to talk to you about is BMP signaling. And this is a bit more of a complex story. The first thing to note here is that there are two apparently conflicting approaches to BMP signaling that can be found in the literature. Older publications, which use knockout serum, reported a requirement for the inhibition of BMP signaling. This is in contrast to more recent publications that utilize better defined conditions in combination with BMP activation. And I'd just like to talk through a few of the newer publications to demonstrate to you the reason for this perhaps confusing situation. A publication by Alan Leung and Martin Garcia Castro in 2016 actually doesn't use any manipulation of the BMP signaling pathway to generate neural crest. However, they demonstrate that if an amount of recombinant BMP4 in excess of just 10 picograms per mil is added to their cultures, then neural crest induction is lost, and instead non-neurolectoderm-like characteristics are induced. And you can see that from the immunos I've lifted from their paper here. If between 20 nanograms and 0.05 nanograms per mil of recombinant BMP4 are used, then SOX10 and PAX7 aren't expressed, and only AP2 alpha, which is also associated with the non-neurolectoderm characteristics, is seen. Similarly, though, if they treated cultures with the BMP antagonist noggin, then neural crest induction is also perturbed. So these two facts together suggest that actually a degree of BMP signaling already exists within the culture that's permissive for neural crest induction. A year later, in 2017, Jason Chu and Lauren Studer published a protocol that utilizes 1 nanogram per mil of BMP4 for the first 2 days of induction, as addition of higher or lower amounts of the protein result in a decrease in efficiency of neural crest induction, as you can see in this graph here. The data presented also demonstrate a fairly high degree of variability between replicates that I would emphasize as characteristic of all of these protocols and something that I'm going to come back to in a minute. In the same year, my colleagues and I at the University of Sheffield published our own work on the role of BMP signaling. We showed that in the right conditions, human pluripotent stem cells respond to BMP inhibition by upregulating central nervous system associated genes like PAX6 and SOX2, while in the presence of BMP4, they instead upregulate non-neurolectoderm associated genes like GATA2, GATA3, and AP2 alpha. It's only in the presence of a moderate degree of BMP activation that neural crest can be induced. Here you can see flow cytometry carried out after 7 days of neural crest induction using a basic and inefficient version of our system. To assess the efficacy of neural crest induction, we used a SOX10 reporter line provided to us by the Studer Lab, which expresses GFP in response to SOX10 induction, and you can see GFP on the y-axis. We use this in conjunction with the surface marker P75, which you can see on the x-axis. SOX10 induction, either seen as GFP expression in this case, or SOX10 transcripts, occurs almost exclusively in cells in the P75 high population. Inhibition of BMP signaling results in a majority P75 low population, and activation of BMP signaling results in a P75 negative to low population. In this basic version of our protocol, in the absence of BMP modulation, we saw a huge amount of variability in neural crest induction efficiency, seen here presented again using flow data. This variability was reflected in the levels of expression of BMP4 and detectable BMP4 protein levels in the culture. We hypothesized that the observed variability in neural crest induction efficiency was due to variable BMP ligand expression within the culture, and we devised a method that we called top-down inhibition to control for this feature. So this approach is in the schematic on the left-hand side, and using this approach, it's possible to control the degree of BMP-induced signal transduction within the cell, irrespective of the amount of BMP ligand production within the culture. So to do this, neural crest induction cultures are saturated with recombinant BMP4 while simultaneously treated with low levels of the BMP type 1 receptor inhibitor DMH1. In these conditions, it doesn't matter how much endogenous BMP is produced by the cells in culture, as it doesn't change the overall saturation level. So this means that the signal is always attenuated to the same degree by DMH1. You can see from this flow data that using TDI, it's possible to achieve very high neural crest induction efficiencies. Western for phospho-SMAD 158 demonstrates the attenuated level of BMP signaling that's required for neural crest induction. And using this approach, it was possible to greatly improve the robustness of induction, as you can see from this graph. So if you're interested in this, you can find more details in the original article published in Stem Cell Reports in 2017, or in an overview article that I wrote for Current Protocols in Stem Cell Biology, which also includes a second approach for controlling the effect of endogenous signaling. So in summary, TGF-beta inhibition, Wnt activation, and an intermediate BMP signal are all required to direct human pluripotent stem cells towards a neural crest fate. But what kind of neural crest are we actually generating? Well, in 2013, there was a publication by von Mieke and Lorenz Studer, the main thrust of which was actually the differentiation of neural crest into melanocytes. However, in the supplemental figures of this publication, they presented data that show that the neural crest they were generating did not express HOX genes and therefore was likely to be cranial in identity. And that's almost certainly true for all of these protocols that are presented in this schematic. However, in the presence of retinoic acid, it was possible, they showed, to induce HOXB2, B4, and B7, the expression of which is consistent with a vagal identity. So you can see HOXB2, B4, and B7 represented in red, yellow, and blue respectively here, in response to 1 micromolar of retinoic acid added on day 2, 4, 6, or 8 of their 11-day-long protocol. Since then, others have used the same approach to generate vagal neural crest and to characterize them further. I've put a couple of examples here on the right-hand side that both use this approach to generate enteric neurons, which in the embryo are exclusively derived from the vagal or sacral neural crest. So now we can add an extra branch to this schematic. Using retinoic acid, we can redirect cells that would otherwise be adopting a cranial neural crest identity towards a vagal neural crest identity. But what about the neural crest of the trunk? Well, on the basis of the expression of Hox genes and other axial specific markers, the degree of posteriorization that can be achieved using retinoic acid appears to be restricted to a vagal identity. Recently, a number of publications have come out that suggest an alternative route for the generation of trunk neural crest is necessary. Before I go into detail about these protocols, I would like to briefly talk about a population of progenitor cells that exist during embryonic development at the nose street border and the caudal lateral epiblast that are responsible for the formation of many of the caudal tissues of the embryo. So in 1993, Linda Gaunt published a paper where she described the existence of these cells, which we now refer to as axial progenitors or neuromesodermal progenitors, that exist in the xenopus blastopore lip and later caudoneural hinge that are able to give rise to both neuroectoderm and mesodermal lineages. Gaunt made this discovery by marking the cells of the blastopore lip using dye and subsequently showing that they contributed to the spinal cord and notochord. And this transfer section from the paper that I have on the right-hand side shows photo-oxidized dye in both of these tissues. 9 years later, Naomi Cambray and Valerie Wilson then confirmed that an equivalent population actually exists in the mouse embryo. And Valerie Wilson and colleagues have continued to publish on this topic, and alongside others, they've shown pretty conclusively that axial progenitors have the potential to form neurolectoderm as well as mesoderm. And they've even published some data that suggests that they can also form the neural crest of the trunk. The second and third paper shown here are lineage tracing studies that show just that. The first using a random labeling method based on a spontaneously activating LAC-Z gene, and the second by using homotopic grafts from a transgenic GFP mouse. I've highlighted this immunofluorescent image from their 2016 paper because it demonstrates an important characteristic of these axial progenitors, which is the co-expression of the neural gene SOX2 and the mesodermal gene brachyury, or T, in a heterogeneous manner. In 2014, a collaborative effort between Valerie Wilson, James Briscoe, Mina Gutierrez, and Anestis Zacharidis resulted in a publication that demonstrated that axial progenitors, or at least axial progenitor-like cells, could be generated from both mouse and human pluripotent stem cells with the application of both FGF and Wnt signaling. The schematic here, taken from their paper, shows in blue a typical anterior neurolectoderm differentiation approach for human pluripotent stem cells using dual SMAD inhibition. In red and yellow is an alternative protocol capable of generating posterior neurolectoderm as well as mesoderm via an axial progenitor-like intermediate. Notably, the second approach presented resulted in the upregulation of trunk Hox genes like HOXC8, seen in the immuno on the right-hand side, that don't actually respond to retinoic acid. You can see that here that in addition to this, the cells that exist in culture after 3 days of exposure to Wnt and FGF express SOX2 and brachyury in a heterogeneous fashion that reflects that of the axial progenitors that exist in vivo. So the top row represents day 3 cells from the typical anterior neurolectoderm seen in blue in the schematic, and the bottom row represents cells at day 3 from the red line in the schematic and the posterior neurolectoderm differentiation protocol. So this paper did not describe the formation of neural crest from axial progenitors. However, a year later in 2015, a publication by Mark Denham did. So this schematic summarizes 4 published protocols for the generation of trunk neural crest, each one of them via an intermediate cell type that co-expresses SOX2 and brachyury just as axial progenitors do in the embryo. Like the previous schematic, I have the year of publication along the top and the amount of time it takes for neural crest cells to be generated down the side. The Denham et al. Paper published a groundbreaking protocol that used both Wnt and FGF signaling to generate human pluripotent stem cell derived cell aggregates that co-express SOX2 and brachyury, as I've said, and could then subsequently be used to generate neural crest. Although expression of trunk HOX genes wasn't actually presented in the paper, the neural crest in question lacked expression of the cranial specific marker OTX2, supporting a caudal identity for these cells. And this is also the first time that anybody published a protocol generating neural crest from this SOX2 brachyury co-expressing intermediate. Since this publication, the same approach has been used to generate neural crest derived sympathetic neurons and chromaffin cells. In 2018, Tom Frith and Anestis Zacharidis adapted the approach originally published and that I showed to you a couple of slides ago in 2014 by Guti and Zacharidis to generate neural crest and to show conclusively that not only were these cells derived from a SOX2 brachyury co-positive axial progenitor-like cell, but they upregulate a whole host of trunk-associated HOX genes and are capable of generating trunk neural crest derivatives. And here you can see HOXB9, HOXB8, HOXC6, none of which are induced in response to retinoic acid. And again, this approach utilized both FGF and Wnt signaling. And in 2019, my colleagues and I at the Garcia Castro lab published 2 papers dissecting the signals required to generate trunk neural crest. Gustavo Gomez's paper published in Development demonstrates that there are 2 peaks of neural crest induction in response to Wnt signaling, in this case mimicked by the GSK3 beta inhibitor Kaya. So the first peak, which in this protocol is at 3 micromolar Kaya, generates cranial neural crest, and the second peak, which is in excess of 8 micromolar Kaya, generates trunk neural crest. And he showed that only in the high Wnt conditions do axial progenitor-like cells form and can a trunk neural crest HOX signature be seen. And you can see the, again, very heterogeneous co-expression of SOX2 and brachyury only in the 10 micromolar Kaya condition in the bottom immuno there. My own paper last year corroborates this work, although we found that in the absence of BSA, the potency of the GSK3 beta inhibitor was much stronger, and the 2 peaks were at just under 1 and 3 micromolar respectively. You can see from these immunos that the co-expression of SOX2 and brachyury is only seen in the higher Wnt condition on the right-hand side, as is the expression of the caudal marker CDX2. The cranial marker OTX2 is only seen in the low Wnt condition. Interestingly, we also identified a role for FGF in this process. FGF signaling has long been implicated in the posteriorization of tissues and utilized in most of these protocols that I've just described to you alongside Wnt to generate axial progenitors and subsequently neural crest. But we found that actually FGF is not required for either neural crest formation or an exit from a cranial identity. You can see that from the top 2 graphs here. If I draw your attention to the left-hand one, where SOX10 expression is represented, on the right-hand side on day 5, you can see that in the control group, which is just the standard trunk neural crest differentiation protocol, and in the PD17 group, which is when cultures were treated with the FGF inhibitor PD17, there isn't a significant difference in SOX10 expression. Whereas in the case of the addition of FGF2, SOX10 expression is perturbed. In the graph on the right-hand side, you can see that irrespective of the FGF condition we use, OTX2 expression, which is an anterior marker, was never induced, and this is only seen at the lower Kaya concentration. So instead, it appears that FGF is capable of modulating the axial identity within the trunk compartment, with low levels of FGF inhibiting HOX expression without inducing OTX2, and high levels inducing HOX expression and high enough levels that is at the expense of SOX10. So with that, I can add a trunk branch to my summary slide here, showing that with high Wnt signaling, we can direct cells towards a trunk neural crest identity via an axial progenitor-like intermediate. And within this trunk compartment, axial identity can be further modified using FGF signaling. And it's actually only with a low level of Wnt activation that we get cranial or, in the presence of retinoic acid, vagal neural crest. So for the last part of the webinar, I'd like to talk to you about some of the things that we can do with our human pluripotent stem cell derived neural crest and present some brief case studies for these applications. So broadly speaking, I think one of the most exciting things about stem cell technology is the prospect of generating cells for future cell replacement therapy. And we could do this for neurocrestopathies. We can also model these diseases in vitro and use this modeling process to aid in drug discovery. Although human pluripotent stem cell derived neural crest are only an in vitro equivalent to the neural crest of the embryo, we can still study their behavior at a cellular level and use this to inform us about how neural crest may behave in the embryo. I won't talk more on this, but it's worth noting that all of the signals that are required to direct human pluripotent stem cells to various neural crest identities and terminal derivatives reflect those of the embryonic environment. Lastly, it's also possible to actually study evolution using these cells, and I'm going to talk a little bit about a fascinating paper that did just this with a focus on facial morphology. But before we get into the case studies, I just want to give you a few examples of the wide array of neural crest derivatives that we can now generate in vitro. So according to Brian Hall, the neural crest differentiates into at least 21 different cell types that fall within the broad categories laid out in this schematic that I'm using from Nicola Douaran's 2004 review on the topic. Melanocytes were first generated by Yvonne Mika and Laurent Studer in 2013, and you can find a modified version of their protocol in my publication last year, which easily generates the pigmented melanocytes that you can see here at the top of the slide without using cell sorting. Differentiation of human pluripotent stem cell derived neural crest into sensory neurons, which you can see here at the top right, expressing peripherin, islet-1 and BRIN3A, has been used as a functional test to identify neural crest since Oz Pomp's publication in 2005. It's also possible to generate Schwann cells, autonomic neurons, and recently chromaffin cells from human pluripotent stem cell derived neural crest. And finally, it's possible to generate the mesectodermal lineages of the cranial neural crest, including osteocytes, chondrocytes, smooth muscle, and adipocytes. A notable exception to this list of derivatives that we can generate are the dentine-producing odontoblasts that I mentioned in the introduction. So unsurprisingly, with this vast array of derivatives, neural crest-related diseases and disorders manifest in lots of different ways, as you can see here. Many of these disorders are associated with mutations that affect neural crest-associated genes, which I've listed below each one. So the two disease-related case studies that I'm going to talk about in a moment focus on familial dysautonomia, which affects the peripheral nervous system, and Hirschsprung's disease, which is caused by a failure of the enteric neural crest to completely colonize the gut. Other notable examples include cleft lip palate, which affects craniofacial formation, as does Treacher-Collins syndrome and Waardenburg syndrome, the latter also manifesting in pigmentation characteristics. Neural crest developmental genes are also implicated in neoplasias, including neuroblastoma and melanoma. So the first case study that I'd like to show you is a disease model for familial dysautonomia that was published in 2016 by Nadia Zeltner and Laurent Studer. So this disease affects the functioning of the peripheral nervous system, and results in a depletion of both sensory and autonomic neurons, as can be observed in the dorsal root ganglia of patients post-mortem. In virtually all cases, this disease is caused by a homozygous point mutation in the IKBKAP gene that affects RNA splicing of the gene. And yet, despite this, the disease can actually manifest in either a mild or severe form. So in this study, Zeltner generated iPSCs from patients with mild or severe forms of the disease and differentiated these cells into cranial neural crest and subsequently into sensory and autonomic neurons. You can see from this graph that both mild and severe cases, in both mild and severe cases, the ratio of wild type to mutant IKBKAP is very low when compared to controls. Despite this, expression of the autonomic neural marker ASCL1 is only affected in severe forms of the disease, or rather in iPS cells derived from patients with severe forms of the disease. And motility of the cells in vitro is drastically impaired in the severe form, but not the mild form. And I should just point out here that the yellow columns marked with S are the two severe form derived iPS lines, and the green columns marked M are the mild form derived iPS lines, with the blue columns being controls. So yes, I should say as well that motility of the cells in vitro is drastically impaired in the severe form, but not the mild form, as you can see in these scratch assays. Differentiation of neural crest into autonomic and sensory neurons in vitro is again drastically impaired in the severe form, but not the mild form. So what this work was able to tell us is that the mediator of disease severity, rather than being an environmental factor, which wouldn't have been carried through to these iPS cells, is instead likely to be some kind of secondary genetic modifier. And in fact, Zeltner was able to identify 3 candidate genes, one of which was laminin beta 4. And I think that this study really demonstrates the power and utility of neural crest, human pluripotent stem cell derived neural crest in modeling neurocrestopathy. So the second case study I'd like to discuss was also published in 2016 by the Studio Lab. And in this work, first author Farhanak Fatahi describes the generation of vagal neural crest by applying retinoic acid to an otherwise cranial neural crest induction protocol in the manner that I described earlier in the talk. So she showed that these vagal neural crest can upregulate the expression of genes associated with an enteric neural crest identity, including ASCL1, FOX2A, and FOX2B, and then subsequently go on to differentiate into neurons with a wide variety of neuronal subtypes, all of the neuronal subtypes that are represented in the enteric nervous system. What was particularly exciting about this work was that the enteric neural crest generated were able to colonize mouse embryonic gut, and to do so better than cranial neural crest generated in the absence of retinoic acid. So this really highlights the importance of generating axial specific identities with our human pluripotent stem cell derived neural crest. Fatahi even showed that when transplanted into the embryonic gut of a mouse model of Hirschsprung's disease, these human pluripotent stem cell derived enteric neural crest were able to rescue the mice in all cases. And finally, I wanted to say that this paper can also act as an example of drug discovery, as the authors also did a small molecule screen to identify compounds capable of rescuing the migration defects seen in EDNRB null enteric neural crest. And they identified a small molecule called pepstatin A, which targets BASE2, which is an acid protease. And this could be a potential therapeutic for the disease. So the final case study that I want to present is concerned not with disease, but with evolution. As the neural crest is responsible for the formation of much of the craniofacial structure, it's no surprise that small alterations in the expression of crucial neural crest associated genes can lead to major changes in facial shape. We can see this reflected in a number of Neurocrest-associated dysmorphisms, such as cleft lip and palate, Treacher-Collins syndrome, DeGeorge syndrome, Waardenburg syndrome. But in addition to this, the altered regulation of these genes plays an integral role in the evolution of facial shape. So in 2015, Sarah Prescott and Joanna Wisoka published on this topic using a cranial Neurocrest induction protocol originally developed by Ruchi Bajpai. The robustness of this protocol allowed them to generate Neurocrest from chimpanzee iPSCs as well as from human iPSCs and identify cis-regulatory divergence between the species that drives differences in tissue-specific Neurocrest-associated gene expression and consequently in facial morphology. So protein coding regions are actually highly conserved between humans and chimpanzees. And so it's been suggested that morphological divergence is in large part due to changes in gene expression levels mediated by alterations in cis-regulatory elements. And the work done in this paper supported this. So the first plot here shows a global comparison of enhancer activity in iPSC-derived cranial Neurocrest within species in red and between species in blue, and demonstrates that a substantial subset of enhancer elements exhibit species-specific bias. It's about 13%. In the second plot here, you can see that many of these enhancers contain binding motifs for Neurocrest-associated transcription factors. They also found that strongly divergent enhancers cluster together in regions that often fall close to genes that we know are involved in face morphogenesis and Neurocrest development. And those include the genes listed here in the bottom right. And you'll notice SOX10, PAX3, and PAX7 key Neurocrest developmental regulators there. So in conclusion, I guess I just want to say that this is a technology that's no longer in its infancy. With a couple of notable exceptions, we can now generate human pluripotent stem cell derived Neurocrest that correspond to most axial identities and are capable of generating the vast majority of Neurocrest derivatives. And there's a huge amount that we can learn and do with these cells, and we've really just scratched the surface. So I'm sure the best is yet to come. And with that, I'd like to thank a lot of people who've helped me with my work on the Neurocrest. Firstly, all of my wonderful colleagues in the Studer Lab, especially Lorenz himself and Yuji Fan, who's an incredibly talented graduate student that works with me on the Neurocrest project. Much of the work that was my own that I showed you today was done either in the Garcia Castro lab or the Andrews lab, and I had a lot of wonderful colleagues that helped me do that work too, and I'd like to thank them, as well as Ana Maria Navarro, who worked with me on the BMP project when she was at the Karolinska Institute. And with that, I'd like to pass back to Teresa and get some questions from you guys.
Thank you very much, James, for a wonderful talk. That concludes the presentation segment of this webinar, and we will now begin our Q&A session by addressing the questions that were submitted throughout the presentation. The first question I have for you, James, is what is the main difference in the timing of differentiation from PSC to neural crest cells? On the different protocols, why are some differentiation times much longer, like the older protocols were?
Yeah, that's a really good question. Sort of perhaps counterintuitively, it seems that the higher density protocols take a bit longer to induce Neurocrest than the lower density protocols. I don't think that means that they are passing through a different state. I think it's more likely that there is just a sort of regulation effect of the rest of the cells in culture. Also, in addition to that, some of the older protocols required purification steps, and this was achieved either manually or with cell sorting, and that added to the time it took for these protocols.
Thank you. I have another question here referring to the induction protocols in general. This person was saying that some protocols use N2 while others use B27. But besides BSA, there are a number of other components found in B27, but not in N2. Can you speak to the observed differences in efficiency between protocols that use B27 instead of N2?
Yes, I wouldn't say that the maximum achievable efficiency is any different between the two. I think that it's easier to study the environment that the cells within the culture are experiencing in the absence of BSA, because BSA interacts with a lot of the components that you add to the media, as I mentioned with Kaya. As far as the other components in B27, I don't think it's clear exactly what role they play, if any, and for that reason I think, as it's clear that they're not necessary because we can achieve neural crest induction with just N2, so in the absence of these extra components, my inclination would be to not use B27 if possible.
Thank you. I have a question here about your own published protocol. They want to know whether you can expand your neural crest cells, and if so, for how long?
Yeah, it is possible to expand the cells, but only at high density. And I think this was first shown by the Menendez publication, which if you look into the details of the protocol, they maintain a very high density and use multiple sort of passages during their differentiation process. Yeah, my advice to you would be to culture at a very high density and to reseed in the presence of ROCK inhibitor. It's also possible to freeze these cells and they will recover with a very high efficiency. So that's an alternative way to sort of continue studies on them for a long period of time. Make a big batch, freeze them down, and use them from there.
Great. Along similar lines, have you noticed any preferences of different cell lines to be biased towards different neural crest axial patterning?
Towards different axial identities? No, I don't think I have. I think, in my experience, all of the cell lines I've used respond to the different cues that I've described and are here on this slide. Certainly I've noticed a bias towards or against certain derivatives. So in my sympathetic neural differentiation for the paper that was published last year, some cell lines produced sympathetic neurons that were tyrosine hydroxylase positive very easily, and some didn't. But it's difficult to say if, you know, that was cell line specific or not, really. But no, I wouldn't say I've noticed a bias in specific axial identities, no.
Okay, thank you. What about, can you comment on any tips for generating sacral neural crest as a subset of the trunk neural crest?
Yes, very good question. Someone's sharp-eyed and noticed that sacral is not in my schematic here. There have been a couple of different things proposed, and I discuss them in the discussion of my paper in 2019, last year. So you should go and have a read of that. But I would say that just extending the amount of time that these cells are subjected to FGF signaling or increasing the concentration of FGF signaling is not sufficient to generate sacral neural crest. There are other signals that are involved in the generation of sacral tissues in general. And I think it's likely that by utilizing these signals, we'll be able to generate sacral neural crests in the near future.
That would be exciting.
Right?
In terms of the neural crest cells that you generate from your protocol, do you have a sense for what the heterogeneity of these cells is and how reproducible that protocol is? Well, I guess you've already commented on the different cell lines, but the heterogeneity of the cells themselves?
Yeah, I would say in terms of axial identity, these populations are pretty heterogeneous. That's probably the exception to that would be the anterior cranial neural crest that we generate, which doesn't seem to have any HOX gene expression. So I would say is definitely pretty homogeneously anterior in character. But after exposure to retinoic acid, or in the case of the trunk neural crest that we generate, there seems to be a sort of quite a lot of variation in HOX gene expression, at least. Otherwise, I would say that we can now, whether cranial or trunk, we can now generate very pure cultures of neural crest. So it certainly used to be that you would have a lot of other ectodermal tissues in there, whether non-neural ectoderm or neural ectoderm. And I think that especially using TDI, it's possible to get really very high efficiencies now. Yeah.
Thank you. We have one other question about growth matrices in culture. So what, if anything, is known about the impact of different growth matrices, for example, matrigel, laminin, or vitronectin, on the production of various neural cell types? And I think they mean neural crest.
Yeah, great question. Obviously, matrigel and geltrex are ill-defined. They're just extracellular matrix produced by this mouse tumor cell line. And so it would be nice really to move on to a better defined matrix. I have run neural crest inductions on vitronectin instead of matrigel, and on laminin 521. And my colleague Anna, who, while she was working at the Karolinska Institute, actually ran this induction on a panel of different laminins produced by BioLamina. And you can certainly generate neural crest on these different substrates. In my experience, it didn't work too well on vitronectin. I don't know if that's a sort of a fundamental problem with vitronectin, or the protocol just would have needed to have been re-optimized. But I know that it did work well on laminin 521. So I would be using that, except it's quite expensive. So on the whole, we still use matrigel. But I don't really see much of a difference between the matrices. And I think certainly with a bit of optimization, you could probably use any of the ones that I just mentioned.
Great. Thanks so much for your perspective. I think that's all the time that we have for today. So I'd like to thank everyone for coming to listen to the webinar today and for submitting your questions, and especially give thanks to Dr. Hacklin for his time and for a great presentation. So thanks everyone again, and goodbye.